Hello, good morning, good afternoon, everyone, and welcome to this new series of OECD webinars on emerging science. Today we are having the first event in this series. And the objective of this series is to invite a few speakers each time to give a talk about their research in relation to a topic of interest for the future of regulatory safety testing of chemicals. So today the topic of the webinar is liver models for induction, clearance, and toxicity. And we have three speakers on the program. We are starting with Steve Ferguson. So Steve is working at the National Toxicology Program as a scientist in the Molecular Toxicology and Genomics group. And he's going to give us a talk about 2D versus 3D models, HepaRG spheroid models. So, Steve, I'm going to hand over the presenter rights to you, and you can start your talk whenever you want. Thank you very much, Steve, for the talk today.
Well, thank you, Anne, for the very kind invitation to share some of our ongoing research and cell model that I'm particularly fond of, and that's these HepaRG cells. And I hope that my screen is now sharing. So
yes.
Before I begin, I just want to remind everyone that I do work for the U.S. government as a part of the National Toxicology Program, and that I have no financial interests to disclose, and that my statements and opinions and conclusions are my own and don't necessarily reflect those of the U.S. government or NIHS. So, substantial progress has been made over the last 15 to 20 years in trying to understand human liver metabolism and drug-drug interactions related to metabolic processes with pharmacological agents. And that's been done primarily due to the fact that human-derived in vitro models have been effective in predicting a series of things. In particular, the sandwich cultured of human hepatocytes that are depicted here in this photomicrograph have been very effective in modeling nuclear receptor and other transcription factors signaling pathways due to their extended longevity and ability to mimic the architectures of parts of the liver lobule. And in doing so, these pathways are able to mimic drug-drug interactions like induction that are often associated with altered pharmacokinetics in clinical trials and in patients. And these models are great in the sense that they not only model nuclear receptor signaling, but they also retain biliary excretion modeling and uptake transport. However, in ripping those cells out of the native architecture and putting them into a more two-dimensional configuration, they do lose about 90% of their xenobiotic metabolism enzymes, including CYP3A4, sort of prototype clearance compound. And they also have other challenges related to high variabilities and technical sensitivities to handling. And they're also short-lived. The best cultures might last for 1 week or so in a sort of standard sandwich cultured configuration. And so because of that, they present particular challenges to the toxicological community as we begin to think about how we can extend these models towards modeling the more broad spectrum of toxicology. And for clearance, in sort of one of the key focus areas of this symposium, we know that the suspension cells, those sort of removed directly from the liver and used immediately or from cryopreserved form, retain essentially comparable levels of hepatic metabolism enzymes that are lost in several hours, but are effective models in predicting metabolic clearance rates and uptake transport as well, as you look at the different processes that you can model in terms of uptake and efflux, between these two models, you can actually do a decent job at predicting hepatic clearance and drug-drug interactions related to induction, and some folks are actually using inhibition. But again, these models, while their metabolic competence is great, are not very effective in modeling toxicology because they're only viable for a few hours, and they're not effective in modeling efflux transport after they've been torn apart from their native configuration. So it sort of takes this sort of hodgepodge of systems in order to make this an effective system to predict liver. And so we'd like to have systems that are a bit more integrated and sort of combine these processes into a single model system that can be used for toxicology with improved longevities. And the bar, however, for being able to use these models is quite low. When you think about the current world of hepatotoxicity screening, it's largely using cancer cells like HepG2 cells, which when you look at the complexities of the liver and the structures that are shown there and have been known for decades, the models that we currently use for this are just don't resemble this at all. And these sort of immortal dividing cells minimally support drug metabolism, transport, and classical hepatic receptor pathway signaling, with the exception of the aryl hydrocarbon receptor, which does work to some extent in these models. And so it's a real challenge to sort of bridge the gap when this is the current standard. And so just implementing some of the current strategies in drug development could go a long ways, and in addition to that, begin to expand to even better models. So when I think about the goals for in vitro toxicology approaches, I generally think about three sort of buckets of utility. So one is the goal is to just improve our understanding of adverse actions of chemicals in humans, and be able to understand that these chemicals are going to have various effects depending on the exposure level and the duration, and being able to begin to model the complexities of this to gain insights about the molecular level as well as the phenotypic outcomes. As we go into the second bullet point, we wanted to begin to try to understand mechanisms of action, and they may be relatively benign or reversible actions, and they may begin to be toxicological actions that have longer-term adverse outcome associations with them. And we want to be able to use in vitro model systems that mimic integrated biology, that approximate the functionally relevant subsets of biology that are critical to be able to mimic that toxicology. And ultimately, those models need to be quantitatively effective in terms of understanding the response dynamics across the various issues that we face: molecular initiating events, metabolic activation, the progressions of response towards more complicated, more long-term phenotypic changes, and again, concentration response. And so that's sort of a key goal there, is being able to develop model systems that allow us to interrogate that. And then finally, not only the model systems need to function properly, but the assay methods need to be able to give us the specificity to see certain pathways, but at the same time, you want something that can globally allow us to characterize a broader spectrum of biological responses, and to have sufficient context in that response to be able to begin to translate that information in a potency-aligned framework. And my hypothesis in this area is that the best models for quantitatively predicting and translating this information are going to be ones that have adequate modeling of metabolic activation and toxicokinetics in vitro. So the media, if you will, is mimicking the plasma metabolic clearance, uptake clearance, and metabolite profiles that are relevant to that particular tissue in that context. And so that's sort of the key here, is to try to get models that are more closely resembling the media exchange with the system that's analogous to the human situation. So before I go too much into sort of our progress in this area, I wanted to give a little bit of background for those who may not have heard about a cell model called HepaRG. And these models were discovered in France at INSERM, and they're commercially provided to the public by Hepatic. And these cells were derived from a female Caucasian patient with hepatocellular carcinoma and hepatitis C. It is not clear that these cells are hepatocellular carcinoma active in the sense that they do not grow in nude mice, but they do somewhat grow in soft agar, and they're generally referred to as a progenitor cell line. And they've also been tested as negative for hepatitis C. And when the cells are passaged for some period of time and differentiated, they begin to sort of hijack the stem cell differentiation tree and will split into two cell populations depending on the culture configuration that's used. These progenitor cells will sort of differentiate into hepatocyte-like cells, and you can see some of those here in this image, these sort of cord-like structures that will form, as well as cholangiocyte-like cells, which are shown in this sort of more flat area compared to the mountain ranges of the primary human hepatocytes that you can see here. And moreover, these cells differentiate into these sort of co-cultures with very strong metabolic activity compared to things like HepG2 cells. This is a particular example comparing hundreds of donor preparations of primary human hepatocytes in sandwich culture versus HepaRG for CYP3A metabolism, testosterone 6-beta hydroxylation. And you can see here, this is around the mean and well above the median of the distribution of CYP3A4 activity in human hepatocyte cultures. They also support hepatic receptor signaling pathways, and probably the most famous hallmark of primary hepatocytes that's never been effectively modeled in vitro is this sequestration of the nuclear receptor CAR in the cytosol that upon phenobarbital induction activation with phenobarbital elicits a translocation of CAR into the nucleus. And HepaRG cells, to our knowledge, is the only cell model that's able to model this. So these are special cells that not only mimic relevant drug metabolism activity on par with sandwich culture hepatocytes, but they also support translocation of CAR and other receptor signaling pathways. They also are functional for transport. You can see here in this image at the top, we've stained for CDFDA, which is a substrate for MRP2, and the metabolite of that is. And you can see that the HepaRG actually in these hepatocyte ranges do polarize, and they begin to create these apical domains, these canalicular bile pockets that will, analogous to primary human hepatocytes, mimic biliary excretion. And so these models are not cholestatic. They're actually taking up molecules, and they're effluxing into these bile pockets, which then dump back into the media in this particular culture configuration. So, and the advantage of these systems is that probably the most important for our program is that they're year over year available. So we have these cells, and we can use them in a sort of consistent genetic background as one of our donor preparations that we look at. They also have, as you can see here, compared to the marked lot-to-lot variation in primary human hepatocytes with metabolism, and this is true for many other endpoints, they're much more consistent from lot to lot. So this gives some significant advantages in being able to have a sort of consistent screen. And their ability not only to differentiate into these cultures, but to actually trans-differentiate and proliferate to some extent for certain assay types like genotoxicity, where you need a cycle of proliferation to manifest micronuclei formation. These are actually a big advantage to HepaRG cells that may be more difficult with primary hepatocytes that have reached, in general, the cells that are isolated or have reached the end of their proliferating states. So we published this paper in 2016. Actually, it came out in 2017, where we created spheroids of HepaRG cells, and Srinivasa Ramagara is a talented postdoc who's now a research fellow on our team that was able to pioneer this action and produce highly consistent spheroids from well to well by simply adding cells and allowing these ultra low affinity plates to aggregate and to form highly consistent sizes across the plates that we make. And we moved to this model because of the challenges that we had with 2D models that I mentioned in the very first slide. And so I want to go into these models a little bit and describe some of the advantages that we've seen and start to talk about the future where we have models such as this that allow us to screen and integrate a more metabolically robust and viable system that would be more suitable to the more complicated questions that are necessary in the toxicology field. One other thing that we're really excited about this model is that they're significantly cheaper. Primary hepatocytes are particularly expensive, and HepaRG are do cost as well to manufacture. And so you can use significantly fewer cells. So one vial can make one 384-well 2D plate, and we can get 12 to 25 plates with a single vial in these spheroid configurations. And so it's an opportunity to scale as well that we think would be more amenable to a commercial setting. So, but before I go into that, I want to talk about why we moved into the 3D, and probably, as I mentioned in the first couple of slides, is the fact that we know that human liver and suspension hepatocytes have comparable metabolic activity, and we know that these models can predict metabolic clearance rates for moderate and high turnover compounds. But as you go to other culture configurations, the metabolic competence spectrum, if you will, is significantly reduced. It's about 10-fold reduced in primary hepatocytes and 2D primary HepaRG cells, and it's another 10-fold reduced roughly with proliferating HepaRG cells, and I'll show you this in a moment. And then IPS-derived hepatocytes and HepG2 cells would be kind of at the lower end of the spectrum. So the cell type and the cell culture configuration matters, and I want to show you this in a plot that summarizes essentially most of my work over the years with numerous colleagues in the drug metabolism field. So this plot represents specific activities for CYP1A2, CYP2B6, and CYP3A4, three major drug metabolizing enzymes in humans. And on the far left is what we consider the bar, the benchmark. And this is the median activity across hundreds of donor preparations of suspension human hepatocytes, so this model here. And so in this case, this is sort of where the benchmark is. And metabolic competence is oftentimes benchmarked to these models, when in fact, this is the quantitative method for predicting metabolic clearance rates. And you can see in the Tox21 world that I've was joined a few years ago, HepG2s, BG1s, and HEC293 cells have very minimal activity compared to this benchmark. HepaRG cells, while very capable of differentiating, if they're grown in this culture configuration, aren't hugely different from the Tox21 cell lines. They do have better CYP3A activity, and that's in this model here. But if you culture them into a differentiated mode, now the cells begin to adapt and change and mimic better an adult liver state. And so now you see about a 10-fold increase in the metabolic activities of these enzymes. And then if you put them in 3D configurations, either in no DMSO or low concentrations, 0.5% DMSO, you see that they achieve levels that are beginning to be comparable to the median suspension primary hepatocyte data. And so this particular sort of context enables us to feel more comfortable that we've begun to have relevant levels of P450 activity, at least on a per cell basis. And so we're continuing to work in this area to see how many of these spheres we need to get a full biomass of that. But for screening purposes, these definitely are in meeting the mark. And not only do they meet the mark in terms of metabolic activity, but they also retain responsiveness to hepatic receptor signaling activators. So omeprazole is an activator of AHR, classical in drug development. Phenobarbital, as I mentioned earlier, is a CAR activator, and rifampicin is an inducer activator of PXR. And you can see here we got robust response with these inducers for CYP1A2, 2B6, and 3A4, consistent with their responses in vivo. So we've been able to not only achieve high baselines, but also further inducibility, which is difficult in the 2D cultures of HepaRG, which require very high levels of DMSO in order to achieve those high levels. And so the three-dimensional context really is taking this to a new level of sophistication that is beginning to allow us to have a more holistic modeling of the liver. So we've also taken pictures of these. Srini's one of the stainings that he did that actually made the cover of Toxi was this MRP2 staining, where he's in this animation you can see various slices of the liver. And so MRP2 is an efflux transporter that is trafficked when hepatocytes are differentiated into these canalicular domains, and lots of reports of these have been coming out recently, and you can see these sort of channel-like structures that are forming within the sphere, and this is evidence of a highly differentiated system, and these networks are much more extensive than what you see in the 2D cultures, likely attributed to the three-dimensional architectures and the more organotypic culture configuration that's produced in these models, and certainly consistent with the higher level of differentiation we see in metabolic activity. We've also done immunohistochemistry staining with these things, and you can see H&E staining and PAS staining consistent with the types of staining we'd see in liver glycogen storage with PAS staining. Poly-CEA and MRP2, as I said earlier, just further indication that we're getting canalicular domains to form, and you can actually see little pockets forming in this particular slice here. One interesting thing we saw as well, remember I told you that HepaRG are this co-culture of hepatocyte-like cells and cholangiocyte-like cells, and in 2D, roughly are at a 60-40 ratio of hepatocyte-like cells to cholangiocytes. Well, in 3D, over time, that population appears to shift, and in fact, in about 21 days, they form what looks like a significantly reduced number of CK19 positive cells and many more hepatocyte-like cells that are largely localized in the interior of the 3D HepaRG spheroids. And this was a little surprising to us at first, and then we actually found Magnus Ingham and Sundberg's group actually showed that co-cultures of primary hepatocytes with non-parenchymal cells did the same type of response with CK19 positive cells. So presumably these are cholangiocyte-like cells or other epithelial cells that are creating this border. And so we think this is real, and we think these are serving in some sort of structural support, sort of exoskeleton type of interaction, but we're not completely sure you know, what this is going to mean in terms of our modeling of liver, but we feel like this is something that makes sense, and it's certainly increasing the proportion of cells that are hepatocyte-like cells within these models. So not only do we need to have a model system that mimics liver, and we think we're really making some progress with these 3D spheroids, we also need to be able to understand what's happening in these systems with assays that are integrated in these systems. And so cell morphology imaging is really essential for us to qualify each experimental run. Not only these sort of phenotypic markers, but also we want to make sure that as the models are produced, that they are grounded in hallmarks of tissue functionality, in this case, hepatic receptor pathways, or highly effective models to do that. And we've grounded these models for these 5 pathways currently, but we've looked at other pathways as well, and they're functioning in 3D very nicely. We also use cell viability assays to provide context to the assay data. Cytotoxicity assays, LDH Globe in particular, has been a very effective assay, very sensitive even with one spheroid in the spent culture media, and so that allows you to move forward with the assays and not destroy your assays for doing other more complicated things like metabolomics and high throughput transcriptomics. And we actually have this has been a major emphasis point for our program, using this TempoSeq platform with the S1500 gene set of about 3,000 sentinel genes to be able to cover a broader biological response base and integrate these physiologically relevant in vitro models with assay systems that help us start to tease apart the progressions of response. And as I mentioned, metabolomics is also another assay system in spent culture media that we're trying to integrate with this approach. And ultimately, using concentration response and 4 or more days of repeated exposures seems to be effective for us to be able to mimic human responses to pharmacological therapeutics and actually discriminate molecules that have been associated with liver injury versus ones that have generally rarely been associated with liver injury. And so we're very excited about this, but it takes a combination of assay systems and model system and concentration response modeling to begin to get the sufficient context to really interpret the progression of responses that's occurring in these types of systems. There's also sort of a tier of information, and so just using cell viability assays, in this case cell titer glow, for ATP depletion, you can see that the 3D HepaRG spheroids with 6 days of repeated exposure had the most sensitive response compared to a single 24-hour dose of to 3D HepaRGs or to 2D HepaRG or primary human hepatocytes in 24 hours. And so this sort of idea of allowing just a simple cytotoxicity assay and multiple days of repeated exposure to give you a sense of whether something's happening or not, going to very high concentrations. And it was very effective. In this case, you see we have drug analogs, trovafloxacin versus levofloxacin, and trovafloxacin in humans was actually withdrawn from the market because it was a liver injury compound, whereas levofloxacin remains on the market. And you can see here just a simple cytotoxicity assay. You probably wouldn't have launched trovafloxacin in these 3D HepaRGs if you had seen that response. Another comparable analog comparison, troglitazone, which was also withdrawn from the market, sees a clear shift compared to rosiglitazone that's only starting to have some response after 6 days of exposure, and it has been associated with some liver injury, but significantly less than from the withdrawn molecule. So these two case studies, just readily using cell viability assays, were able to distinguish these molecules, and looks to be a useful tool in just rapidly understanding whether there's a liability here for liver injury. Meanwhile, molecules like aspirin and gemfibrozil did nothing in the assays, even out to 6 days exposure. And so we think this is a good start. We've also moved into this high throughput transcriptomics research, and we in fact have now published this approach. In this particular case, we started with the 2D HepaRG, and so this is giving you kind of a global picture of what's happening with our high throughput transcriptomics platform. So on this axis, you can imagine each sort of differentially expressed gene is accumulated in an integer fashion. So the lowest concentration, benchmark concentration that's observed gets a 1, and so on for 2, 3, all the way up to very high levels. And what you can see is that, as you might expect, the number of differentially expressed genes goes up as you have higher associations with liver injury. And so you can see acetaminophen, aflatoxin, chlorpromazine, menadione, troglitazone, benzalpapyrine, trovafloxacin. These are all molecules that are producing in very high numbers of differentially expressed genes across the concentration response. And on this axis are the actual respective benchmark concentrations for each gene level response. And what we did in this paper was we actually were able to show that the concentration at which a molecule crosses this threshold, which we defined with a subset of molecules that are rarely associated with liver injury, we were able to show that the potency at which you cross this threshold is tracking relatively well with the potencies at which you begin to see liver injury in humans. And you can see acetaminophen on the far right, whereas environmental toxins like aflatoxin and benzalpapyrine showing up on the far left, as we might anticipate. But this, we've proposed this as a quantitative approach and an untargeted approach to be able to predict liver injury, and we're now extending this approach into the 3D spheroids as well. But even in the 2D, it did a nice job in getting us the ability to discriminate these molecules with the high throughput transcriptomics. Moreover, once you've seen a response, you can begin to look at the pathways that are affected by these various molecules, including those case study comparisons of trovafloxacin and levofloxacin, which is down here in the weeds, and be able to see some ability to discriminate between those processes and understand what is being changed, and begin to get a sense of mechanistically what types of mode of action might be useful for further investigation. And here is just a blow up of the trovafloxacin versus levofloxacin gene level BMC curve, so you can see again this similar to this cell viability assay was able to readily distinguish these molecules from one another. And since these are both pneumonia medications, the human effects would presumably be off-target effects, and so you might imagine not wanting to launch this one that's changing tons of genes when you already have this one on the market. So we're feeling comfortable that the model system is starting to give us some utility, and the assay system is giving us a window into this, and in context with cell viability assays gives us a really useful tool to begin to quickly interrogate chemicals and to bend them in these sort of moving averages as we continue to grow the numbers of chemicals that we're looking at. Another example that was really exciting for me, because I'm really concerned about metabolic activation of toxicity, is cyclophosphamide. It's notorious to require metabolic activation, particularly CYP2B6, to produce primary phase 1 metabolite that then goes on to produce phosphoramide, mustard, and acrolein. And in order for this chemotherapeutic to have its action, it requires this metabolic activation. And so in the paper we published, 2D differentiated versus non-differentiated HepaRG were compared, and as I showed you earlier, the non-differentiated have very poor metabolic activation, including low expression of CYP2B6 and much higher expression in the 2D differentiated. And what you can see here is that we were able to readily distinguish in the 2D differentiated lots of pathways, many of which are actually related to therapeutic observations. DNA damage, cell cycle, hypoxia in elderly patients has been shown, responses to glucocorticoids, and immune system processes, as well as apoptosis. And these pathways make sense, and there was sort of a progression of responses towards more toxicological as you went across the concentration gradient. But notice that in the proliferated cultures, we had very little pathways observed, and that's consistent with their dramatically reduced metabolic competence. And so we think that a more differentiated model system gives you the ability to mimic metabolic activated processes, but also begin to model a more complex diversity of biological processes that mimic closer the tissue responses. And when you think about this in context with the Cmax, so the therapeutic plasma concentrations of cyclophosphamide are roughly 120 micromolar, and you're starting to see this inflection point near those concentrations, and that actually makes sense when you think about chemotherapeutics, that you're trying to maximize the effectiveness and minimize the amount of overt injury to the body. And so it actually makes sense that you would we would see this inflection at concentrations approximating that. So not only qualitatively but quantitatively, we're beginning to have hope that these types of models can provide a predictive and translational ability with unknown, untested chemicals, including those in our environment. So considerations for the broader application. When you think about the benefits of 2D and 3D HepaRG compared to HepG2 and other sort of classical cytotoxicity screens, it's really important to understand that the totality of function is for a tissue model is particularly essential when you're trying to have a better predictivity. And so this superior functionality and longevity gives you the opportunity to look for things like chemical accumulation effects that might be going on, and more complex, more toxicologically relevant responses to chemical exposures. And so we believe these models are relatively easy to use, they're relatively inexpensive, they are reproducible and consistent, and they have superior functionality for improved predictivity. I think we're beginning to see that. The types of chemicals that we've tested so far have been largely reference chemicals. We have had a few environmental chemicals, as I showed earlier, benzopyrene and aflatoxin. We have a broader set of around 600 chemicals that are in the current pipeline to run in these types of screens. But our primary objective here when developing a new paradigm was to start with drugs, including failed drugs, with some prototype environmental chemicals, because this is where we have human data. And if we're trying to model human toxicity, we need to have chemicals that we've tested in humans and have some anticipation. And so that's the rationale so far, but we will absolutely be extending that into environmental chemicals in the very near future, and we've already got some of that work underway. In terms of gaps and challenges, our biggest issue with the 3D spheroids is imaging. We believe that phenotypic anchoring of these systems is going to be critical, and as they progress towards adverse outcomes, we want to be able to see examples of, say, steatosis or fibrosis, various apical outcomes that we believe imaging is going to be essential, and so we need improved methods for imaging 3D spheroids. In 2D, they do a very nice job, but they just simply don't model, from what we can tell, as organotypic of a response as we'd like to see. We also know that our models currently need non-parenchymal cells, Kupffer cells, stellates in particular. Models are emerging out there now that use these routinely, but there aren't great commercial sources for stellate cells in particular that would allow you to look at donor variation. Kupffer cells are a little bit more mature, but still have their challenges, and it's not clear at this point whether using non-parenchymal cells from different donor preparations leads to different levels of response. I would imagine as we get better resolution on those responses that's going to be an issue where we might want matched donor preparations. But for some of the more broad responses, the Kupffers and stellates mixed with different donor preparations from donors that were used for human hepatocytes seems to be doing a decent job, but it is a challenge to get these into the HepaRG cells and then figure out if they're modeling the system analogous to primaries. We also need to do a better job in characterizing metabolism and transport in the 3D spheroids. As I mentioned earlier, we want the TK to model and reflect human plasma profiles of clearance and metabolite formation. And so we need to consider the biomass of a single spheroid and maybe increase the number of cells and reduce volumes to get closer to the allometric scaling of humans if we hope to have an improved quantitative translatability of our potencies. And then finally, transferability and economic aspects. The cell viability assay with 3D spheroids, in my opinion, is ready to go now. If people wanted to get a sense of that, it's a very simple assay to run. The plates are available, the cells are available, and using that type of assay system is pretty straightforward and can be standardized very quickly, in my opinion. As we ask more questions of these systems and we're asking them to model a broader set of responses, the assay systems like the transcriptomics to give us that sense of responses and tuning in the maturation time and the culture media to allow those pathways to be proven to work properly and functionally, and identify major sources of variation that help to tune these systems, including endocrine pathways related to metabolism of glucocorticoids and the cortisol levels that are used in these systems. They're going to be things that we need to continue to work on and improve. But as we ask increasingly complex or broad questions of these systems, they simply need to be calibrated to add that in, and I think they have the potential to do that very quickly. And then finally, in 6 to 12 months of additional qualification and tuning, I think we'll be ready for full-scale implementation in the field. And so it does take a village, and in particular, I want to acknowledge Srinivasa Ramagari, who's done a lot of the seminal work in this area. A new postdoc in our group, Caitlin Lavridge, is beginning to tackle the maturation and the conditions of HepaRG cells. Scott Auerbach and Rick Paulus for their work with the transcriptomics and helping us get that moving along, and Mike DeVito, our branch chief in the laboratories, for leading and inspiring our group to continue to push towards toxicologically useful systems. And thank you for your time.
Thank you very much, Steve. That was an excellent presentation and introduction to the topic of liver models for future regulatory applications. And thank you for giving a talk that I think will be within the grasp of the broad regulatory regulators in chemical safety testing area that are interested in these kind of talks. And with that, we'll continue our webinar with other presenters. Thank you.
So first of all, I would like to thank the OECD and especially Anne Gourmelon to have invited me today to speak in this webinar. I'm currently in a postdoctoral position at the ESPCI, the School of Physics and Industrial Chemistry in Paris, and I'm working with a new 3D liver cellular model called Hepatotoxicity to validate it for different applications such as drug-drug interaction or hepatic clearance and also hepatotoxicity. So currently there are two big groups of 3D culture methods, scaffold-based culture methods and scaffold-free culture methods, on which I will focus right now. In this kind of culture, you have first the hanging drop and the force floating methods based on the self-aggregation and the self-organization of the hepatocytes, either in a hanging drop or in a surface specifically treated so that the hepatocyte can't attach on it. The biggest advantage of this technique is that you can precisely control the size of the spheroid that will exclusively depend on the number of cells you add. But if you add too many cells, a necrotic core will appear as the oxygen and nutrients can't reach the core of the spheroid. This technique has also other disadvantages. First, the handling of the spheroid is quite difficult as you just have one spheroid per well or per experimental point. You need to be careful when you change the medium to not aspirate the spheroid. Moreover, as it is a naked spheroid, pipetting it could disturb or disaggregate it because of its fragility to mechanical stresses. Also, this method is not adapted for high throughput as you have just one spheroid per experimental point, and you need to pull at least 8 or 10 of them to have enough cells to run your assay. The quantity of cells limited also your readouts depending on what you want to see. Another method is based on the agitation of a big quantity of cells inside a bioreactor. While this allows producing a huge number of aggregates, you can't really control the size of them, and there will be heterogeneity of the size between the different spheroids inside the bioreactor. Moreover, this system has other common disadvantages with the previous one. It's difficult to handle naked spheroids, and you need to pull some of them depending on the assay you want to run and the readout you need. Finally, a third system consists in the embedding of cells or spheroid directly in a bead of alginate. In this kind of system, the alginate allows protecting the spheroids against mechanical stresses and makes them really easy to manipulate. Nevertheless, this system is for the moment still an in-house system and not commercialized, and it results in a heterogeneity in the size of the aggregates and in the number of cell spheroids per beads. So we were looking for a model that can combine a maximum of benefits and obviously a minimum of disadvantages of the system I've just talked about, and we found the hepatotoxicity. So the hepatotoxicity have been developed by a startup company named Vitro using the BioPearl proprietary technology, and are now cellular model commercialized as a plug-and-play solution. Hepatopearls consist in encapsulated primary human hepatocytes in an alginate shell. These primary human hepatocytes could either be freshly and newly isolated from a liver or cryopreservated. Around 200 cells are encapsulated inside each liquid core capsule, and in this microenvironment, they will self-assemble and self-organize into 3D spheroids with a diameter of around 150 micrometer, as you can see on this picture. This process of spheroid forming takes approximately a week. And with this specific concentration of cells leading to this specific size of spheroid, there is no appearance of any necrotic core, as you can see on this picture. It's a classical live-dead assay where living cells are green and dead cells are red. And you can see here that a large majority of cells are green. The alginate itself has multiple properties. Extracted from brown algae, this natural polysaccharide is able to polymerize and to form a porous and permeable shell in this kind of system. Due to the size of the pore, which is around 20 nanometer, the alginate shells allow oxygen and nutrient exchanges while allowing scaffold 3D microtissue growth. On the contrary, big molecules as antibodies, bacteria, or also immune cells are unable to go through the shell. In a clinical context, this property combined with the biocompatibility and the non-immunogenicity of the alginate itself is really interesting to protect the spheroid from immune reaction, for example. Finally, the BioPearl technology allows by modifying the encapsulation system to modulate either the size of the capsules or the thickness of the alginate shell, depending on the cellular type to encapsulate and the wanted application. So before I'll show you some example of application of the hepatopearls, I'm going to detail some characteristics of this model by comparison with the gold standard primary human hepatocytes cultured in monolayer. So first, as you can see on this slide, the hepatopearls are viable for more than 6 weeks, by contrast to the classical 2-day culture onto collagen 1 coating, where most of the cells are dead after 3 days. In hepatocytes, you can see that after encapsulation, as I mentioned before, it takes approximately a week to form a real compact spheroid, characterized by a really thin black border. And these spheroids keep their morphology and their viability, as demonstrated by calcein-positive staining, for at least 5 weeks. It means that you can extend the duration of your experiment with this kind of model, or test repeated or chronic exposure of a compound, and even envision repeating your experiment with the same batch of hepatopearls some days or weeks after your first experiment. If we are looking at gene expression levels, you can see that hepatopearls is a physiologically relevant liver model. They indeed express different genes of the CYP450 superfamily, such as CYP3A4, CYP2B6, CYP1A2, and CYP2C9, but also liver-specific nuclear receptor as a pregnant X receptor, constitutive androsten receptor, and aryl hydrocarbon receptor. And also phase 2 enzyme UGT1A1 and UGT2B7. These expressions are maintained all along the lifespan of the hepatopearls at levels generally higher than those of 2-day hepatocytes. And this temporal consistency during 35 days allows going for chronic studies for at least 5 weeks. We can notice here a huge decrease in the relative expression of some gene, especially in the CYP1 at day 4. this is linked with the formation of the spheroid that occurs during this specific period. Moreover, synthetic activities of albumin and urea follow the same profiles as the gene expression, quite high during the first week post-encapsulation and a decrease in the following week to reach a plateau. At this step, the concentration of these two compounds are still higher than the ones from 2-day culture, and they are maintained for at least 4 weeks. Finally, the hepatopers mimic the liver architecture as demonstrated by immunostaining. You can see here the staining of zonula occludens 1, which is a membrane polarity marker present at the tight junction between the hepatocyte, as well as the staining of the filamentous actin, which is also a polarity marker. Moreover, the hepatopers are able to specifically excrete the carboxyfluoroethane diacetate. This compound is specifically processed by esterase in the hepatocytes, where it will become fluorescent. It will then be excreted by the multidrug resistance protein 2 in biliary canaliculi, and this is this structure that you can see on this stack. So after having shown some characteristics of the hepatopers model, I will now explain different applications where they have been used. So cytochrome P450 enzymes are essential for the metabolisms of many medications. Although this class has more than 50 enzymes, 6 of them metabolize 90% of drugs, with some of the most significant enzymes being CYP3A4, 2B6, and 1A2. Genetic variability in this enzyme may influence a patient's response to commonly prescribed drug classes, including beta blockers or antidepressants. Cytochrome P450 enzyme can be inhibited or induced by drugs, resulting in clinically significant drug-drug interaction that can cause unanticipated adverse reaction or therapeutic failures. So the knowledge of the most important drugs metabolized by cytochrome P450 enzyme, as well as the most potent inhibiting and inducing drugs, can help to minimize the possibility of adverse drug reaction and interaction when a patient takes more than one drug at the same time. So to check the possibility to use the hepatopers in this search of potential CYP inducer or inhibitors, we have, in collaboration with Sanofi, incubated the hepatopers with non-reference inducer referenced by the FDA. We measured the respective cytochrome activities for 6 weeks. And on these graphs, you have a bar from clear to dark for each week by comparison with the 2D hepatocyte in gray. And as you can see, all along their lifespan, the hepatoper present an inducible CYP activity for each cytochrome with a dose-response effect. For the CYP3A4, we can also notice a hepatotoxicity at the higher dose, where the activities are weaker than these of the 2 lower doses. So the hepatopers can be used to look for potential CYP inducer or inhibitors and possible drug-drug interaction through this kind of method. The liver is the most important organ for the elimination of a drug. It is thus essential to know whether a drug has a high or low hepatic extraction ratio, because in a clinical context, it determines dosage adaptation, especially in patients presenting condition altering hepatic blood flow or hepatic enzyme activity. So hepatic clearance quantifies the loss of drug during its passage through the liver, and it results from hepatic metabolism and biliary expression, and it is a function of the hepatic blood flow, the drug plasma protein binding, and also the activity of liver enzymes and transporter. We then use hepatopers to assess the clearance of a well-known drug, the midazolam, in a specific pathway with first hydrolyzation by the CYP3A4 enzyme before a conjugation by an enzyme of the UGT family. So still with Sanofi, we use liquid chromatography tandem mass spectrometry to analyze the metabolism of the midazolam and the appearance of the first and the second metabolites at different time points during 48 hours. So as you can see on this graph, midazolam is hydrolyzed as soon as it is in contact with the hepatopers and has completely disappeared after 24 hours. The first metabolite is immediately conjugated to form glucuronide 1-OH midazolam, and after a peak of concentration at 2 hours, its concentration slowly decreased, while the one of the second metabolites increases in parallel at the same range. So this experiment demonstrated that phase 1 and phase 2 enzymes are functional in the hepatopers and that they can be used for clearance application and especially for low clearance compounds. Drug-induced liver injury is a cause of acute and chronic liver disease. And as the liver plays a central role in transforming and clearing chemicals, it's susceptible to the toxicity from these agents. So certain medicinal agents, when taken in overdoses and sometimes even when introduced within therapeutic ranges, may injure the liver. More than 900 drugs have been implicated in causing liver injury, and it is the most common reason for a drug to be withdrawn from the market. So these are the reasons why we use the hepatopers to test if they are a good model for hepatotoxicity purposes. In this experiment, we used a pool of 3 different donors to ensure the heterogeneity. We treated the hepatopers with 4 drugs with different known hepatotoxic potential for acute, so 24 and 48 hours, and also chronic toxicity, 13 days. Interestingly, we showed that the hepatopers can categorize drugs according to their hepatotoxicity potential, and above all, that they can be used to detect toxicity due to chronic exposure, as it is the case for chloramphenicol or diclofenac. So to conclude, we can say that the hepatopers are a new model physiologically relevant and closer to in vivo, easy to handle, and which allows running chronic exposure assays. With its high rate of production and the possibility to get customized plates with a precise and predetermined number of capsules per well, this model seems adapted to high throughput activities. Finally, it's economically interesting as it allows using a less important quantity of cells to perform assays in drug-drug interaction, hepatic clearance, or hepatotoxicity areas, as I shown you before. So to finish, I would like to thank all the members of our team at the LCMD, Laboratoire des colloïdes et matériaux divisés, and also CPRIO for the authorization to use some of their picture, and obviously our collaborators at Sanofi, and I thank you all for your attention.
Thank you very much, Jérôme, for this very nice presentation and for sharing all this information with us today. Thank you.
Good afternoon. I'm Dan Tagle. I'm Associate Director for Special Initiatives at NCATS at the NIH, and I'm here to talk about microphysiological systems, particularly focus on liver on chips for drug safety and efficacy assessments. And so just to provide a brief outline, I'm going to speak a little bit about the National Center for Advancing Translational Sciences, or NCATS, and then switch into a general overview of what microphysiological systems are, and then dive deeper into the liver on a chip, and then integrated multi-organ on chips, including the liver, and then building confidence on the microphysiological systems, and provide a brief summary of my presentation. So the National Center for Advancing Translational Sciences, or NCATS, was established in 2012 by the United States Congress, essentially to address the central issues that are preventing advancements in drug development. So it has a central mission of catalyzing generation of innovative methods and technologies that will enhance the development and testing and implementation of diagnostics and therapeutics across a wide range of human diseases and conditions. So many of you might know that the current time, the average time to develop drugs takes in the order of 10 to 15 years at a cost of $2.6 billion. And the current drug discovery paradigm has a failure rate of 90%, 55% of which is due to lack of efficacy, and 28% is due to toxic effects in humans. And so what we really need are better predictive tools that will assess for the safety and efficacy of various drug candidates. So a number of studies have shown that the current paradigm that we're using in terms of both 2D cell culture systems as well as in vivo animal models are not predictive. A more recent study shows that the highest predictive rates or true positives can occur in dogs, primarily in cardiac and GI conditions, and rats are also predictive only primarily for renal and respiratory conditions. And this is looking at a study of 3,290 approved drugs with over 1.6 million adverse events over the course of 70 years. And so what are microphysiological systems? So this is the program that NCATS established in 2012 to address the issue of the problems in drug development. So microphysiological systems are essentially to create an in vitro platform that uses human cells and tissues and combine that with technologies, advances in technologies in stem cells, microfluidics, and bioengineering, essentially to create in vitro devices that are better able to predict safety and toxicity issues from candidate drugs. So shown here in the picture, these microphysiological systems are representative of the circulatory, the endocrine, the GI, the immune, skin, musculoskeletal system, nervous system, reproductive, as well as respiratory and urinary system. So it's essentially the major organ systems in the body, and these in vitro platforms are engineered so that they are physiologically relevant and that they also can inform genetic diversity, and of course has to be relevant in terms of representing disease pathology. It needs to be modular in the sense that it could be plug and play, kind of in some ways kind of like Lego blocks, and at least be viable for 4 weeks to allow for chronic drug testing. So this is what a typical organ on chip or microphysiological systems would look like. So essentially there are multi-channel 3D microfluidic cell culture chips that stimulates the activities, the mechanics, and physiological response of entire organs and organ systems. So they essentially would have scaffold, cell structure, spatial temporal patterning, perfusion, bioreactors, innervations, host responses, and functional readouts that would allow it to mimic human conditions. So for the scaffold, essentially purified ECMs, cells are primarily made up of primaries or induced pluripotent stem cells. Structure, meaning the porosity, topography, and stiffness of the organ is mimic. Spatial and temporal patterning has controlled release of cytokine and hormone gradients. Perfusion, because these are now 3D constructs, there needs to be a microfluidic cell representation of vasculature and ability to flow nutrients and oxygen into these 3D constructs through either vasculature or microfluidic channels. In the bioreactors itself, biomechanical properties are represented, and then signal propagation, coordinated response for innervation, generalized inflammation and specific immunity for host responses. And we're also trying to develop real-time, label-free, non-destructive sensing capabilities to do functional readouts, and that would include imaging, and then being able to take in vitro and correlate that to in vivo through systems integration and multi-scale modeling. So just to show how to mimic a human organ function, so this would be the lung, human lung. So essentially, the human lung is broken into its basic functional element. In this case, the alveoli or the air sac, as shown here. And this is a rendering of what an organ on chip or a lung on a chip would look like, where human lung epithelial cells, shown here in blue, are on the airway side, and then separated by a thin membrane would be and human endothelial cells on the vascular side, and then channels on each side of the device is subjected to negative pressure and cyclic vacuuming, and that allows the membrane to stretch and also stretches the human lung epithelial cells as well as the endothelial cells. That allows it to mimic this stretching function that allows the human epithelial cells to secrete surfactants. So this is a rendering of the microvasculature on a chip, where you can see fluorescent beads coursing through it, just as you would see in blood vessels. This will be a gut enteroid that has been innervated and showing peristalsis, the movement of the gut enteroid muscles. And this would be the beating of a human ventricular chamber, showing the not only the contractility but actually actual pumping capability of this system. There are many engineered platforms shown here. So this is actually the human lung on a chip. This is the microvasculature on chip, which is this. And this would be the GI system, liver, and kidney are represented in this Nortis device. This is an example of what a integrated system would look like. This happens to be the female reproductive system that contains female ovary, human uterus on a chip, the ectocervix, and the fallopian tube on a chip, and that's coupled or linked up with a human liver on a chip to indicate how female hormone changes during the monthly cycle can affect drug metabolism. And this is another integrated system that has the skin, the human liver, vasculature, and muscle and bone. So as I've already stated, NIH through NCAT started this program in 2012, and it was a 5-year program that ran through 2017. The first 2 years of the program was to develop the bioengineered platform as well as the cell resources, primarily primaries and induced pluripotent stem cells. And then the last 3 years of the program involved functional validation. That means being able to show that it mimics physiological response, and that's primarily done through a training set of compounds. This were obtained through collaborations with AstraZeneca, Pfizer, and GSK. And this training set of compounds are about 100 or so compounds that have gone through preclinical safety assessments using the current 2D and in vivo models, and have then gone on to human trials, but have been subsequently being withdrawn due to adverse events. So the question would be whether the chips developed would actually predict the human response that were not predicted by the conventional ways of assessing safety and efficacy. NIH also partnered with a number of institutes and centers at the NIH as well as with DARPA and from the Department of Defense. And DARPA was primarily interested in developing a 10-organ platform, as shown here, and then also collaboration with the FDA. That is a non-financial partner, but essentially provides regulatory insight and expertise in terms of how to develop the systems that would be used for regulatory decision making. So over the course of the 5 years of the program, more than 500 publications came out of this program that's been published in a number of high-level journals. But we also wanted to shift gears and be able to tell you a little bit more about the liver on a chip. And so the liver on a chip, one of the examples I'm going to be showing was the one that was developed at the University of Pittsburgh with the PI as Lance Taylor, a number of collaborators here. And so in this particular case, the self-assembly of hepatocytes and non-parenchymal cells, and this is using the Nordis chip. So in this case, it's not just hepatocytes, but you also have non-parenchymal cells such as Kupffer cells, endothelial cells, and stellate cells represented in this system. And this is what the Nordis platform looks like. This is where the cells reside, and as shown here in this blow up, that's where you actually get the hepatocyte cords being formed primarily through self-assembly. This shows the importance of having the co-culturing of hepatocytes and non-parenchymal cells. So if you just have hepatocytes shown here, over the course of 28 days, you see actually a decreasing level of expression of albumin, whereas if you start adding NPCs, non-parenchymal cells, not only do the albumin level goes up, but it also is expressed or secreted at physiological levels. The same goes for LDH expression. It goes up during the course of 28 days as you also increase the complexity of co-culture systems, as well as also in the enzymatic activity of CYP3A4 becoming more physiological the more MPCs are represented in the system. This slide shows the importance of having fluidic flow into the system, and so shown here in red would be a co-culture, meaning the hepatocytes and non-parenchymal cells co-culture together, but under static conditions. And then the blue is shown with fluidic flow and showing that you get better albumin secretion as well as urea secretion with flow, and you also get bile canicular export with flow compared to a monoculture with just the hepatocytes. So you get better formation of the bile canaliculi. So the liver on a chip is also able to show fibrotic response. So when methotrexate is administered, the stellate cell gets activated and express collagen 182, as shown here with a fibrotic response. And then you can also elicit immune-mediated hepatotoxicity by lipopolysaccharides, as shown here, with increasing concentration of LPS and showing also the increased level of TNF-alpha release from the Kupffer cells. And moreover, if you couple LPS with trovafloxacin, you can see immune-mediated toxicity as shown here in blue compared to just the trovafloxacin by itself without LPS, shown in red. As I mentioned, one of the goals of the program is also to develop non-invasive functional readouts. And so in this case, optogenetic-based lentiviral systems were incorporated into sentinel cells. That would be indicators. So essentially, the change in fluorescence would indicate various cell processes, whether it be cytochrome C release, reactive oxygen species generation, or caspase-3 activation. And so just as an example here, when the liver on a chip is treated with benadione, which is a known liver toxin at 100 micromolar concentration, you can see a real-time change in the cells going from red to green, indicating apoptosis, and this coincides with a decrease in mitochondrial membrane permeability, as well as an increase in radical oxygen species generation. You can then look at a number of compounds to see how well this system, the biosensing system, performs. So like if you look at trizodone, which has no known liver toxicity and it's cleared through the liver quickly, you can see that the various cell readouts are all in green, indicating that there's no damage to the liver cells. Whereas if you look at the menadione, which is a known liver toxin, ROS, calcium uptake, mitochondrial membrane permeability, apoptosis, and bile efflux are shown either in red or in yellow, indicating liver toxicity in this particular case. This has now been then used to determine mechanisms of toxicity for a number of compounds, whether it be controls such as caffeine, where you don't see any toxicity, to known hepatotoxins, where you see changes either in albumin, apoptosis, bile efflux inhibition, fibrosis activation, as shown here by the arrows, LDH, ROS, and immune-mediated toxicity, and also looking at drug-induced liver injury, as shown here in this list. This is just to show how the liver on a chip is also increasing in terms of complexity, and it's evolving to be much more sophisticated. So this is the self-assembly liver on a chip that I just showed, with primary hepatocytes and then the non-parenchymal cells, and then the readouts using biosensors, as well as the albumin and urea indicators. You can also develop, it's now evolved to a liver acinus model that has primary hepatocytes and primary endothelial cells, and using cell lines for the stellate and Cooper cells, to the current state, which is now a vascularized liver acinus microphysiological systems that has iPS-derived hepatocytes, stellate cells, cholangiocytes, and primary sources for Cooper and leukocytes and monocytes. And the biosensors are also gene-edited iPS lines. This just shows the coupling of the liver chip with a number of other organ systems. In this case, showing liver hooked up to the kidney, to the gut, and blood-brain barrier, with a cardiopulmonary assist showing pulsatile flow into the system. And this is showing the importance of how the coupling of the organs on the chips is important in terms of determining secondary metabolites. So in this case, looking at aristolochic acid nephropathy. So shown here, if the kidney on a chip was treated by aristolochic acid by itself, there's no toxicity detected. But when aristolochic acid is flowed through the liver and then into the kidney, then you see cell death happening in the proximal tubule of the kidney. and this is caused by the metabolite, which is the sulfated conjugated form of uracilocleic acid, as shown here. So if you just have the vehicle, this is what that looks like when treated. When hooked up with the liver, this is what the cell death looks like after 24 hours, and then you can block this by treatment with probecenid, indicating that this sulfated form is taken up by the kidney using one of the OAT transporters to which probecenid acts, showing that the liver is the one that produces the sulfated forms that can then induce kidney toxicity. So this is a general slide showing what the current state of the NIH tissue chip microphysiological system looks like. This is actually now a consortium of a number of stakeholders in the field within NIH, pharma, other government agencies, academic institutions, but I'll focus primarily in the rest of my presentation with where the state of the art is in terms of developing tissue chips for disease modeling, as well as for validation of the system through the testing centers, the TCTCs. So this is a slide showing all the different chips being developed now for disease modeling for various conditions, anywhere from cardiomyopathies all the way to lung disorders, polycystic ovarian syndrome. So there's a number of projects that are looking at liver and chip multisystem pathologies, and also looking at, for example, type 2 diabetes, where the liver on a chip is hooked up with the pancreatic islet cells, with adipose tissue, or with a GI system, as a way of multi-organ integration to model type 2 diabetes. As I mentioned, NCATS have been working with a number of stakeholders, and that includes the IQ consortium, in this case made up of 20 pharmaceutical companies that essentially comprise of a multidisciplinary team of scientists from pharma, and that is being able to leverage their expertise and to be able to inform NCATS as well as the FDA in terms of what would work best for pharma and in terms of adoption of this technology in terms of the drug development pipeline. And out of the interactions with the FDA and with the IQ consortium, NCATS formed a validation framework for microphysiological systems, and with the first level of validation being physiological, meaning the tissue chip developers would essentially replicate organ function and structure, and as I've already mentioned, validate it using a training set of compounds. The second stage of validation is the analytical, and this is done by independent testing centers, and essentially testing for robustness, the reproducibility, reliability, and relevance of the chips, and this is with a validation set of compounds, and this is done informed by the IQ consortium and the FDA. And the third stage of validation will be the industrial stage, wherein industry and regulatory agencies have actually started using these chips on their own with a proprietary set of compounds, particularly in a CRO type environment. And we think that these three steps are key to the eventual adoption and commercialization of the tissue chip technology. Just to give you a sense of what the independent testing centers are doing, so these are, as I've said, established to create independent sites of validation and it's informed by the FDA and IQ Consortium. So the testing centers currently are housed at Massachusetts Institute of Technology, where Murat Chirit and Alan Grotzinski are the PIs, and then Texas A&M University, where Ivan Rusin is the PI. And these two testing centers are going to be testing all these chips in the first 2 years, and these have all been tested since they were funded in 2016, and all the data as well as the best practices and protocols are being housed at the database that NIH set up at University of Pittsburgh, where Mark Sherdock is the PI. The first publication on the independent testing of the kidney on a chip was published in Nature Scientific Report that just came out late last year, and that's shown in this reference on the bottom of the slide. This shows the increasing commercial activities around organ on chip technologies, with a number of companies that have spun out or been formed around organ on chip technology, whether it be from looking at organoids to actual organs, integrated organs on chips, such as Hesperus and tissues, where you have multiple organs, anywhere from 2 to 10 organ systems being coupled for drug testing. And then certainly in the last couple of years, there's been growing pharma and FDA interest in the use of organs and chip technologies. The FDA, for one, has formed partnerships with two of the spin-out companies, Emulate and CN Bio, to actually bring the technology to the FDA. They also had a recent public hearing on what it takes to promote the development and use of new technologies to predict human response. And then Johnson & Johnson and Merck have also partnered with Emulate to bring in that technology in terms of drug development pipeline, as well as AstraZeneca have also partnered with Emulate in this particular case to also actually set up an organ on chip center in one of their sites in Sweden. And just to summarize where the organs on chip would fit, so as you go into the drug development pipeline, going from target development to preclinical as well as clinical stage, looking at target-based development and evidence building, you would see that spheroids and organoids would essentially fall within the realm of having the capability of high throughput screening compatibility, but decreasing physiological complexity, whereas the organs on chips are certainly much more of a low throughput to medium throughput technology, but certainly has increasing physiological complexity and can be more deployed into the late stage preclinical and early stage clinical development. And to summarize, the tissue chips or organs on chips are much more physiologically relevant, and because it reconstitutes the microarchitecture of the organ systems, has tissue-tissue interfaces, precision flow control, perfusion and innervation of the tissues, dynamic and biomechanical cues are reproduced in the system, as well as spatiotemporal chemical gradients, and has the ability to have circulating immune cells, and as well as other capabilities to look at secondary metabolites. And with that, I'll end the presentation and just show that the tissue chips are currently being used for predictive testing and safety assessment, as well as for disease modeling, and NIH is in the process now of deploying this technology also for clinical trials on chips, as well as for precision medicine. And with that, I thank the audience for listening in, and thank you very much.
Thank you very much, Dan, for this very nice presentation and for the wealth of information you kindly shared with us today. and also for having told us a bit about the way these systems are validated for future use in a regulatory environment for drug registrations and evaluations in the US. So thank you very much